All right, let's go ahead and start with sort of the introductions. First, welcome everybody to the May version of our Craighanine Science webinar. We have, I think, over 200 people that are on the email list, and over 100 have signed up for today's webinar, and we're just really thrilled by all the interest in these topics. You can see that we have scheduled for June. That's going to be June the 4th. Will be that first Friday of the month. Ben Rook and Yu-Chun Kao will be speaking, and we'll be advertising their talk in the coming weeks. So a little bit of etiquette. Please keep yourself on mute, at least during the presentation. But one of these things that was really successful last month is we had fantastic discussion after the talk. So when the talk is over, we hope that you'll submit questions either via chat or pop your camera on, raise your hand. And both Julie and Mark have graciously agreed to stick on after the talk. And we don't have a hard 1:30 cutoff. You know, probably, we won't hold them on too much past 1:30, but if the discussion is good, you know, we'll continue the conversation as long as they're willing. So with that, I want to introduce two esteemed scientists who, if you follow Craighanines or just Great Lakes in general, these two need no introduction. Kicking it off will be Mark Ridgway from the Ontario Ministry of Natural Resources and Forestry. And then Julie Turgeon from Université Laval will pick up sort of the mid part of the presentation. And then Mark Ridgway will bring it home in the end. And they think that it's about a 45-minute talk, but I'm sure it's going to go really fast because I know it's going to be really interesting what they're going to be talking about based on their research in the Algonquin Park and more broadly, I expect across sort of North American Craighanines, ecology and evolution. So let me stop sharing my screen, give Julie a chance to share her screen. She's going to be controlling that. And with that, I think we're ready to rock and roll. Carrie, most people are in so far.
Yep, we're getting a couple of stragglers, but most people are here.
Okay, great. Julie, that's the full screen that we can see. So, yeah, looks great. Excellent. So Mark, take it away. Thanks again, you two.
Okay, thank you both for the invitation. Thanks to everyone who's here today to listen to our Cisco stories in Algonquin Park. We think it's quite fascinating, and I hope you do too. Next. Yeah, so we have, Julie is controlling the slides, so we minimize going back and forth. So I will from time to time signal Julie to switch slides. Julie and I had the privilege of being part of a great team of young people doing this work over a set of years, about 6 or 7 years in its total duration. Alan Bell, Gabrielle Payette-Lozier were our graduate students. The funding was from the Great Lakes Fisheries Commission. And we had a team of technicians who kept the trains on time, kept them moving, kept everybody going, and a small group, actually fairly large group of summer students over those years that were essential for us. The program is basically a lake netting program, which the students, I think, grew to like, and then flying among the lakes, which the students immediately liked. So it's a pretty cool project, and I think for them it was the summer job of a lifetime. Okay. So let's begin with something familiar, the Great Lakes Craighanines. And here's an example, a poster that I think most people know. From Lake Michigan plus Ardidi makes 7. The other Great Lakes, of course, had versions of these or replicates of these, but maybe not in that such a number. Okay, this is a well-known group of fish, including the demise of these fish and the current problems that are facing the restoration. They're a focus of attention for a very long time, actually well over a century. The first indication of the diversity of these fish were published 7 years after Darwin's Origin of Species. There is a long and confounding taxonomic journey over decades and decades of work. It's quite a journey. I'm not going to go into detail, but I will highlight that in 2016, a group of Great Lakes scientists who work in this area essentially came together to help resolve this and published a review essentially calling all these Cisco forms. So for the people who are listening here who may not be Cisco heads, you will hear us sometimes you might hear Cisco people referred to as Zinithecus or Hoyai, when in fact those are now sort of quick labels. I'll call it that. We see them all as forms, and we'll use the word form through this presentation. But I might revert to old talk and say a Zinithecus type or a short jaw type, for example. And the Great Lakes, for a long time, were considered a source of Cisco diversity. You can tell by the way people wrote decades ago, and the way they named things across the landscape. And so we also raise a question here whether it's actually better described as a case study of Cisco diversity. And we ultimately then, through the 2016 review, land on Coregonus artidae sensu lato, broadly defined, to capture the species complex. Now, inland lake Cisco diversity is also well known, of course, and has been for some time. And northern lakes back in the 40s actually started labeling things like the Great Lakes. And now, of course, we know that there are cases of ecological speciation or island evolution, if you want a quick phrase. Okay. So when we think about ecological speciation in northern fish, what do we see? Well, we see, or we need, an ecological opportunity gradient, something for forms to develop. And mysis has been proposed as part of that gradient by Randy Eshenroder and colleagues over 20 years ago. Niche expansion has to occur with adaptive region. It has to occur. In order for other forms or diversity to develop, niches have to expand, or the world has to expand for these organisms. Morphological differences are usually associated with this, and for fish, it's quite often feeding structures, because the niche expansion for fish in these cases is often trophic niche expansion, and we believe that's the case with Cisco. There's also, I call this niche conservatism or the phenotype environment correlation. Essentially, it's the replication of these forms across different islands or lakes, so that lakes of similar habitat can produce similar forms. You get this, so you get this replication, this kind of conservatism to what might develop. And of course, then there's genetic differences, and whether or not they're more related within a lake than among lakes. But some of the things that Julie's group has uncovered on the genetic differences are so interesting. It also questions topics around the origin of these fish. So today, we're going to talk about Algonquin Park, what we found in the park, and its wider implications. So here's the park in Southern Ontario. I have a map here of 3 of the Great Lakes to help orient people in case you haven't been or know about Algonquin Park or been to the park. If you've been to the Adirondack part in upstate New York, you're literally on the same landform as Algonquin Park, so you have an idea what this place looks like. Now, the park itself is fairly high elevation, as you can see on the map, but except for the northern part of the park, it's down lower than 381 meters, and that's an important piece. Okay. So I'm going to briefly go through a set of maps here about deglaciation. It's very difficult to talk about ciscos without providing a context of glacial retreat. So the top map is the full glacial extent, and there are fish refuges around them in the hatched area. The one in the middle, the 2 M's and a question mark, is the Mississippian and the Missourian to the further west. And the question mark I have there is really my inability to discern the boundary between these two. I know there might be one, maybe somebody has a better idea, but it's not certain to me. Throughout these slides, Algonquin Park is the red point on the map. The next 6 panels, reading one row down to the next, so go left, right within a panel, then left, right in the next panel, are a series showing Arthur Dike's maps of glacial retreat from the early 2000s. So let's begin in the upper left corner. Lake Agassiz, AG, develops as the headwater of the Mississippi River system. And if you go to the right and then down to the next row and then down to the next row, you'll see Agassiz growing through glacial retreat. And then finally joining in the lower right corner, another lake, Barlow Ojibwe. Agassiz and Barlow Ojibwe were joined in the end, and that drained through the Winisk River. Then let's return back up. Lake Algonquin, proto-Lake Huron and proto-Lake Michigan, is also growing through this series of maps. But around 13,000 years ago, when the park was becoming free of ice, Lake Algonquin drained through the park for many years and essentially lowered the level of Algonquin. So then to the right of the middle row, at about 12,000 years, what was Lake Algonquin were remnant lakes. And then finally the ice retreated and the Great Lakes formed as we see them as the Laurentian Great Lakes. Next. So this story in the park occurs in the top part of the park. Here are the lakes that we surveyed. Red and black lakes are within a valley system bounded by the red dotted line. You can see the red dotted line through there from the west at Foss Mill to the east. Red and black lines are mysis-based lakes. The Lake Algonquin drained through here, leaving behind a zooplankton community reflective of the Great Lakes, and that includes mysis and fish. Blue lakes are above the flood or inundation level of Algonquin Park. They do not have mysis, and they have Chaabris as the primary food web. Each of these lakes was surveyed with stratified random nets on the bottom, the North American Standard gill nets set for 1 hour. And pelagic nets were set for about 4 hours, either midday or in early evening, never overnight. There were no overnight sets here. The Black Lakes, oh, sorry, Joy. The Black Lakes are lakes where Cisco formed Blackfin, or what we call Blackfin. Red lakes are monomorphic lakes, but with mysis. Blue lakes are monomorphic ancestral Cisco, but with Chaabris. Next. So this is the full distribution of the Cisco survey in the park. I'm sorry, this is the full species range, the native species range. So we associate with Cisco introduction to this landscape, associated with the drainage of Lake Algonquin. This figure shows nicely the delta fan in the east on the right, which was the delta for the draining Lake Algonquin. Foss Mill in the west is the entrance point, and the water moved cascading through that system of lakes in the valley, out through that delta to the Champlain Sea to the east. In this, okay, next, Drew. Okay, so here's the lakes with the Blackfin. I'm just sort of taking lakes off the map to make things a little clearer. First, to the west of this park that you can't see is a lake called Mimi-Sagamessing, or in the indigenous language, Lake of Many Bays. That's part of our analysis later on that you hear about, but it's west towards the shore, towards the coast of Lake Huron. White Partridge Lake is a lake that Julie and others wrote about in 2016 about the question of Xenithicus being unique to these lakes. And so White Partridge has the Xenithicus form or the short jaw form of whitefish, not Cisco, not white, not Blackfin. Next. Okay, 2 guys and 2 Ciscos. This is the Cedar Lake example, just to give you an idea of how big these fish are. They're 30 to 40 centimeters total length. They're quite sleek and robust looking. And this was one day process, part of our Cedar Lake survey. Next, and then here's some more images. Radiant Lake, one of the 3 Blackfin lakes with ancestral Cisco. Radiant Lake, then Cedar Lake, and then Hogan Lake. And these images portray very clearly why we call them Blackfin. It's quite dramatic to see a fish come up with these kinds of jet black fins. The colors on these fish actually coincide closely with Kels's descriptions of luminescent purple and blue on the side of Blackfin. These match very closely just in their appearance what Kels wrote about. So, yeah, so it raises questions, doesn't it, about the origins of these. So here is a zooming in now in that valley system that has Hogan and Cedar. Hogan and Cedar drain to Radiant. So Radiant is receiving water from those systems. Cedar and Hogan were separated by about 12,000 years. They have long ago lost contact with each other after the Lake Algonquin subsided, but they do drain to Radiant. So we'll begin by indicating here the Gillraker distribution. All Cisco talks must have Gillraker distributions. And we do. The left is the ancestral or pelagic normal Cisco, 5 to 10 years, and easily twice that for what we call the Blackfin, and they get up into the 60s. Radiant does not have a clear dichotomy between the Blackfin and Ardidai on the left, so it's a skewed left Gillraker distribution. We model their habitat distribution. It's 15 to 30 meters. It's deep. It's not the deepest part of the lake, but it is hypolimnetic. 5 to 8 degrees is the temperature in which they reside during the day. Thank you. Okay. Next, Julie. Okay, so this is where Julie will take over for a bit.
Okay, thank you, Mark. So as Mark just mentioned, Cisco diversity is not restricted to the Great Lakes. The same forms are also present and still being discovered in inland lakes throughout the range. And the ideas about the origin of these forms or species has evolved through time. Just a second. So the first idea was pretty simple. People imagined that each and every species of the Great Lake had survived in a Mississippian refugium, that they colonized the forming Great Lakes, and that some of them afterwards dispersed throughout the range. So that was pretty classic thinking at the time. But meanwhile, some people were starting to propose that in fact, there was a single original Cisco species and that it invaded from the Mississippian refugium again in the Great Lakes, and that adaptive radiations occurred within each of the Great Lakes afterward, and that one lineage dispersed through the range, and that independently there was the evolution of similar forms and patrick forms in many other lakes throughout the range. Then came the idea that there might be an Atlantic lineage in ciscos. This they resulted from a thorough comparative analysis of fish distribution in Ontario, a fossil that was found in the Champlain Sea, and the first genetic data gathered around James Bay and Hudson Bay, which all concur in suggesting that a second lineage might exist and that it could have dispersed from an Atlantic refuge. Since then, I carried a few studies over a few decades with mitochondrial DNA, microsatellite, AFLP, and now SNPs. And this picture here describes what I think has become sort of the new model of Cisco evolution. Of course, it could change again. And we show here that there is indeed two lineages. One is presumably from the Atlantic, the other from the Mississippi. There is a zone of mixing of these fish in the Great Lakes and in the area once covered by Barlow Ojibwe. There is a break in distribution between Nipigon and Superior, and to the west, we found the Mississippian lineage. And also, I would say that for most of the data that I've gathered, the idea that there was independent origin and divergences in different lakes across the range seems to be the best explanation to explain why there are so many forms. So back to the topic of Blackfin, where are they? So historically, Blackfin, or Nigripennis as they are called in the Great Lakes, were present in Lake Huron, Lake Michigan, Lake Superior, probably, and Lake Nipigon. And inland, Nigripennis or Blackfin were unknown of. And nowadays, the Nigripennis has disappeared from all of the Great Lakes proper, but it remains in Lake Nipigon, which will be our reference, the sole reference possible. And we say that they are present in Algonquin Park. So given the location of these new Blackfin population in Algonquin Park, a question readily comes to mind, which is, did Blackfin colonize Algonquin Park from the Great Lakes, or more exactly from Lake Algonquin when it was pouring through the Fossil outlet? So if this is so, and given the admixture of lineages in the Great Lakes, we would expect the Blackfin in Algonquin Park to also carry this genetic signature of admixture. To assess this hypothesis, we analyzed fish from areas where we know of only a single lineage. So you know the color code here. So the blue for a Mississippian reference, where there's only a single lineage. In Lake Champlain, where there is only the Atlantic lineage. In the Great Lakes with the mixed lineage. And we also included Nipigon since it is our reference for Nigripennis. So the idea was that we should find the same thing in Algonquin Park. Second genetic signature is expected. So if Blackfin population, and here you see my icon, Blackfin, Blackfish. If Blackfin colonized through the Fossil outlet the area of Algonquin Park as a distinct lineage, it should also form a distinct lineage in the lakes of Algonquin Park. So here you have the code, black for Blackfin, and these little pointy fish here for Cisco. So I will present just the two main results quite rapidly, and then I will discuss each of them. So unexpectedly, we did not find a mixture of two lineages in Algonquin Park, but we found only one lineage, which we say is from Atlantic origin. So we found the mixing again in the Great Lakes and the signature of the two reference. And this incidentally was done with a new set of marker, 6,000 steps. So it doesn't seem that the Blackfin originated from the Great Lakes with this raw result. The second signature was that the genetic structure was by lake and that Blackfin population did not form a genetic group. For those who are not used to this type of graph here, basically genetic groups are indicated by color. So you see here the black box, which represent different lakes. And you see my little icons. So the Blackfin here and the Cisco here, they're in the same lake, and then they're in the same genetic group. So the Blackfin here, 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 and here, and here do not form a single group. Otherwise, they would all be in the same box with the same color. So the Blackfin belong to this green Atlantic lineage. We expected to see mixing in Algonquin Park, but we observed only one lineage. And the signal is pretty strong and pretty clear. So back to why we expected the mix. It's because we think that there are two lineages from two distinct refugia, that they came into Lake Algonquin, and when it poured into the Fossil outlet where Algonquin Park now stands, we should have this mixing there. And instead, we didn't see the mix. So one explanation is just that something was blocking the access to the area, but this seems pretty improbable and even silly, given all the evidence there is that fish and tons of biota came from the proto-Great Lakes through this area, let alone the mysis that is defining the signature of the Great Lake waters going this way. So why would Cisco not go through is pretty improbable. So what happened? Well, you should say just so what, who cares? But some people care about these stories about where the fish came from. And what happened? we decided to use the end money of the grant to genotype a few more samples, which we position in critical areas, namely areas that could only be reached at the in the main phase of Lake Algonquin when the water level was very high. And we did the same thing for the east, where it could only be reached through the Champlain Sea. And also we had some fish from Amanda Akis and Wes Larson from, what are the states there? Wisconsin and Minnesota. So this is what we found. So this is the new fish we genotyped, but also all the fish genotype I had for 20 years. So that's about over 3,000 fish. And we sort of found a way to put all of this together. And we see the same general signal of two lineages, a break between Nipigon, mixing in the Great Lakes, in the area of Barlow-Ojibwe, and in the new areas that we included, we see only the Atlantic lineage. In areas reachable by the Champlain Sea, in area of high water of Lake Algonquin, and in the latest outlet of Lake Algonquin. And incidentally, and surprisingly, on the west part of Lake Michigan here. So this distribution around here was also found by Amanda Akis and Wes Larson. Here, the data that they shared with me is results from an analysis using trees. So you cannot see a mix in there, but they do see the two clades, and the break in between the distribution of the two clades are totally coherent with what I have found with this new data and with previous data. So how do we explain this new distribution, this global distribution of lineages in ciscos? And more precisely, what do I want to explain is the lack of mixing in the area of Algonquin Park. So we have a new proposition to the effect that there was no fish in the Mississippian refuge, and that instead they dispersed from a Missourian refuge and an Atlantic refuge. So I'm just going to sketch very roughly how this could have happened. So we would have the early dispersal from the Atlantic refuge into the Great Lakes and into the area associated with the Champlain Sea. Then, when the fossil outlet poured east, this lineage would colonize the Algonquin Park area. And meanwhile, fish would get access to early Lake Agassiz from the Missourian refuge. Then the fish would spread across the vast proglacial Lake Agassiz and Mekano and come back toward Nipigon, while the eastern lineage was moving north in Barlow Lake. And the last phase would be the mixing of lineages in the Great Lakes and in the Barlow-Ojibwe area. So this colonization scenario, I think, is plausible. It explains the eastern distribution of the green lineages that is completely in accordance with Atlantic origin based on traditional zoogeography and comparative phylogeography across many, many species. And as for the origin from the Missourian refuge, there's not so many studies using genetics that have detected a Missourian refuge, but those that have show a very similar distribution for this blue lineage here. So this is for walleye, and you see circled in blue the area that was colonized by what was inferred to be a Missourian lineage, and it's really similar to what we found here, including Nipigon, and here on the at the eastern point of current extant Lake Superior. Also, the Missourian refuge is inferred for fish that have distribution going far northwest and in western Hudson Bay, which is the case for Cisco. But it's not perfect as a scenario, and there was another hypothesis that was proposed by Eshenroder very recently, to the effect that there is no Atlantic refuge and several Mississippian refugia. So the dispersal towards the Champlain Sea is quite likely possible. If the fish could come from Champlain to the Mississippi, they could go from this in Mississippi to the Champlain. And this scenario also makes it possible for the mix of the lineages in Lake Agassiz and Ojibwe and in the Great Lakes. However, I see a few problems with this scenario. First, there is no genetic evidence that has ever been found for 3 lineage or sub-lineages in the area of the Mississippian refuge. And second, if Cisco from the Mississippian refuge could reach the Champlain Sea and expand in all the areas associated with the Champlain Sea, then we could think that many other species could have done this. Hence, arguing against the Atlantic refugium amounts to dismissing all previous evidence for the signature of an Atlantic lineage in the Eastern Range. And finally, this hypothesis supports early mixing of lineages in Lake Algonquin, and therefore it doesn't explain why we don't see this mix in the Algonquin Park, which is the focal point that I'm trying to explain today. So we will need more data, as always. So we will need very good data that will be able to distinguish the 3 proposed lineages if they exist. And my guess at this point is that we will find something that will make both scenarios partly true, but we'll see. So back with Blackfin population that are in Algonquin Park, these Blackfin populations do not form a genetic group. And this structure that we see here by lake, with the 2 farms being more closely related within lake than across lake, is really the signature of in situ divergence of the 2 farms. The interesting thing in Algonquin Park is that we are in a relatively small area that you could portage in 3 or 4 days, I guess. And it's a very pristine area. And we see very variable degrees of differentiation across lakes. We see that for the genetics. So we have genetic differentiation based on 6,000 SNP only in 2 lakes. We have ecological differentiation, but sometimes one form, like Blackfin or Cisco, is sort of caught in the wrong habitat. And we also have a varying degree of morphological differentiation that is truly parallel. So as expected, if you have repeated evolution along the same adaptive gradient. So here are 3 graphs, and each is about 1 morphological trait, Gilbreaker number, linear traits, and shape. For each graph, we have on the left here, the variation for that trait in lakes where there is only 1 ecotype, 1 farm, only the Cisco. And we have in the box here, the values for pelagic and benthic fish, Blackfin and Cisco, in lakes where they are sympatric, with the lines relating the 2 sympatric forms. So the starting point is likely the Cisco morphology that is found in allopatry. And you can see that indeed the pelagic fish resemble, although not totally similar, Mark will talk about this, to the allopatric Cisco. And then if you put this line in these sympatric, if you compare the sympatric form, you can see that the end point, where the arrows point, is always in the same direction across lakes. So that's the real manifestation here of parallel phenotypic evolution. And interestingly, the differences are similar to what we see for the Artidae nigripennis pair in Nipigon, for which we didn't have the shape, but for the Gilbreaker and the linear trait, we say that it goes in the same direction. So it's the same, but at the same time, it's not to the same degree in all lakes. In the end, I think we can sort of see a continuum of the divergence in ecology, morphology, and genetics in this lake. So I've draw a very personal graphic figure here for you. And I believe that this sorts of recapitulate the stages of ecological speciation in this very small area. So we have lakes with and without mysis, and only pelagic cisco. So you would have like a 3 miles lake with no mysis, Grand Lake with mysis, but only cisco. In lake where mysis exist, we have cisco-looking fish that are found in both habitats, such as in Lake Cochon. And a similar situation has been documented by Peter Jacobson in Minnesota, where it was shown that cisco-looking fish are actually eating invertebrate, benthic invertebrates. Then we have lakes where we have two morphologically differentiated forms, which are different in shape or in gill raker. And this would be the example of Mimosygomacing for the shape and Radiant for the gill raker. So this is probably where selection comes in, with shape and gill rakers distinguishing the forms. And ultimately, we have sort of the last stage where differentiation is very pronounced for all traits, and genetic differentiation is significant. But again, in all of these lakes, sometimes we find one morphotype being genetically or just morphologically differentiated in the wrong habitat, like in the water column or in the benthic habitat. So this is my way of putting all this together, and it, I think, underlies the very important aspect of ecological opportunity and how it affects the morphology of fish. And I will leave this subject to Mark to finish our talk.
Okay, thank you, Julie. The next slide is building on this mysis argument. As I said before, Randy and co-authors over 20 years ago wrote about mysis as probably an agent or an important element for driving cisco diversity in the Great Lakes, particularly at depth. And so that's actually kind of one of the beginnings of the mysis hypothesis. Kells noted the importance of mysis as well to the forms in the Great Lakes. So if the mysis hypothesis is true, its particular role and as an agent or as an ecological gradient, then we ought to see an expanding trophic niche pattern in some way. And it's almost always, as I said before, on feeding and food web elements. So using carbon and nitrogen from mussel in each of these lakes, C is chaobarous systems, M is mysis system, and using Nitrover for standard ellipse area. And the chaobarous systems on the left are rather small. And as you go to the right in all of the mysis lakes, the standard ellipse area increases, so their trophic niche is expanding. Now, in Manitou and Grand Lake, they have pelagic forms, as I indicate on this graph. And if you held them in your hand, you couldn't tell them apart from catfish or Carl Wilson. And they're single phenotypes. In 3 Mile and Cauchon, as Julia alluded to, they have a behavioral type. We're catching fish on the bottom and on the top of Cauchon, and you couldn't tell those cisco apart either, although when you do numerical analysis, there's some difference. But if you're in the field, they look the same. So you have a single phenotype, but in those cases, they have a behavioral or habitat expansion. And then finally, we have the 3 lakes with real blackfin, even if Radiant doesn't have a separate form. And so each one of these steps along the way from pelagic single to benthic pelagic and mysis systems is expanding the niche, trophic niche area of cisco, and sometimes by a factor of 2 and 3. Hogan is an unusual lake in many ways. We can talk about that at the end of the talk, but nonetheless, it's part of that story. So this is something that absolutely has to happen in some way, whether you're looking at cisco, anolis lizards in the Caribbean, or any other species groups that's radiating. It's got to have a bigger world. Next. So looking just at Cedar and Hogan now with the 2 clear forms of cisco and blackfin. These are carbon-nitrogen ellipses. And the multiple red, multiple black ellipses are to register the plausible location and distribution of the niche, carbon and nitrogen trophic niche. Now, what's interesting here is the ancestral Cisco in both cases. Actually, the ellipses in both lakes look really, they kind of look different one to the other. But what is interesting here is the overlap of the Blackfin with the ancestral form is nearly identical, although they look different. The overlap is nearly identical, about a 46% chance of overlap of the Blackfin with its ancestral form. And the ancestral form has a 70-some% overlap with its derived form, the Blackfin. So this is a case showing the Blackfin niche is different and moving in a similar way away from the ancestral niche of the red or the ancestral Cisco. And it also indicates a higher trophic level by virtue of the black is tending to be higher. And in fact, that's true. They are a higher level because they are mysis predators. This is a story that was worked out years ago by Joe Rasmussen and Jake van der Zanden. And these are actually some of the lakes that are part of that publications back in the late 90s. So mysis is inserting a trophic level here, and you see it in the way the black ellipses are relative to the red ellipses. Next. So here is something that I'm working on with Alan Bell currently, not submitted, not published. Gill rakers. So this is one. Okay, so here's one of my favorite little topics of discussion: Cisco people and European lake whitefish people, Corriganine people, always publish gill raker count distributions. It helps determine the presence of different forms. But gill rakers are really sieving apparatuses, aren't they? And so what we've done is count the gill rakers on the lower gill arc and express it as rakers per millimeter of lower gill arc. The upper gill arc is not useful, even though it has gill rakers, because where bone and cartilage begin and end is difficult to discern. So by staying with the lower gill arc, we're staying with two clear bony structures that have a clear beginning and have a clear end. On the left are Cisco, single morph Cisco populations in Algonquin Park from either Chaobarus in blue or Mysis food webs. Single phenotype, typical Cisco, different food webs. And there is the amalgamated box plot for the distribution of raker density, rakers per millimeter. As you go to the right, you're into the special forms that we've been talking about, including all the way to white partridge on the right. And what you're seeing here, we're interpreting as character displacement. This is way more useful than just counting gill rakers, and I'll show you why. All the upper boxes are the low gill raker count Ardidae form. But when expressed as raker density, when expressed in a way that's closer to the sieving function of the gill rakers, they're actually highly dense gill rakers. The bottom set of boxes are the higher gill raker count, Blackfin form or almost Blackfin form, or in the case of white partridge, the Xenithicus form. They may have high gill raker counts. Actually, white partridge has a low gill raker count. But the Blackfin may have a high gill raker count, but they have a very low density relative to their ancestral form. And those ancestral forms are higher than the lakes that are in blue and red on the left. They're pulling apart, so to speak. Now, in White Partridge Lake, that is a very low gill raker count and is really, actually in terms of this story, is really quite an extraordinary species pair. So you have a Xenithicus-like bottom form, and you have a pelagic-like Ardidae ancestral form. So this actually started us thinking about the allometry. And one of the things that was published years ago, actually Louis Bernatchez wrote this in a chapter in a book, indicated that when you have an absence of Cisco, lake whitefish has an opportunity to develop a small form. And white partridge is an example of that. It has no lake whitefish. So I'll show you what we mean by this allometry story. Next slide. So here is the fork length of Cisco and the fork length of whitefish. I'm not going to go into the lake details. These are all in the park. These are classic, normal, ancestral whitefish and normal pelagic artidae. The artidae are solid lines, and the whitefish are dotted lines. Red is for mysis food webs, blue is for chaoborous food webs. So mysis, cisco actually are quite parallel in slope, that differ in elevation. They have very common types, so to speak, if you think of the ancestral ones, when you're looking at raker density as a function of length. Lake whitefish, not so much. It looks like it's a size effect, but this is the ancestral types for these two food webs and these two species. Now, in the next two slides that come up, they will be gray background. So let's look at white partridge here. So white partridge, Cisco, the solid line at the bottom, is actually grouping allometrically with a whitefish form. These are whitefish mimics. This lake has no lake whitefish. This is entirely a flip story from what Louis Bernatchez wrote about. So in lakes without whitefish plus Cisco, they're pretty rare. This is what can happen. They're actually moving towards a whitefish form. They're functional whitefish allometrically. We didn't do a good job on getting pelagic forms. We didn't get enough, so I just simply plotted the data. This is just the data. We need to do better on that. But anyways, the spectacular story to me is that white partridge, benthic Cisco, the Xenithicus type, groups with whitefish ancestral forms. Next slide. And then in the middle of the park, we haven't talked about this lake. It's called Big Trout. It's right about in the middle of Algonquin Park, and it has no Cisco. Cisco were blocked from penetrating this watershed by a waterfalls, small one. And so the whitefish that were there didn't have Cisco coexisting. And this is a classic whitefish species pair. There's the large benthic bottom type, and there's the little Cisco mimic pelagic type. And so the pelagic type lake whitefish and big trout, the dotted line still groups with the whitefish allometric story, the allometric axis, if you want to think of it that way, but the slope of that line is identical to the slope of the Cisco allometry story. The benthic or normal lake whitefish is grouping down perfectly settled in with the whitefish allometric story. And this is exactly what Louis wrote about in the chapter. So this is a case where you can get a Cisco mimic and you can show that it's a Cisco mimic, at least functionally allometrically. Now it's not as good a sieve as the Cisco, but its pattern is the same. So one of the things that we're thinking about this, and we're just thinking about it, is that these allometric patterns, there's a difference in how they respond. The Cisco elevation can change. That's how you can get white partridge settling down with lake whitefish. And they're much more amenable or much more malleable in this pattern as an elevation story. And the lake whitefish are not so malleable in their elevation story, but there tends to be a lake whitefish axis. Anyways, this is all literally, we're writing about this and going back and forth. Next slide. So to wrap up, what do we see in northern ecological speciation of northern fish? We see we have to have an ecological opportunity gradient, and we believe that that is mysis. And I'll just add here that we know that mysis diversity and behavior and distribution changes with lake depth. So we think this is a lake depth story, but it is most definitely a mysis story. We see niche expansion with adaptive radiation. Mysis makes Cisco food webs, mysis makes Cisco trophic niches bigger and bigger. Next. We see morphological differences. We've gone through the gill raker stuff, the raker density, the shape analysis, and so on. So there's a whole suite of things that are related to either propulsion or actual filtering and sieving that easily, fit the classic story. And we see niche conservatism or phenotype. I mean, as interesting as this story is, there is still only an outcome of two types. Now, without whitefish, white partridge showed its two types. It had a whitefish mimic instead. But there is a replication then among these lakes or islands, given just how much diverse can you be, in other words, given the size of the habitat. And then the genetic differences really, at least in our minds, realigned thinking for us regarding the origins of Cisco in the Great Lakes, and as well as the more local and valley specific story of the drainage through the top of Algonquin Park. So, thank you. And we would love to discuss this stuff till the sun goes down.
Thanks very much, Mark and Julie. Fascinating talk. I looked up and I couldn't believe it was already 1:20 because it went so fast, like I suspected. So yeah, so everybody can use their little reaction clap to show your gratitude. And hopefully we can either use the raise hand function. I can see the participant list. Hopefully the raised hands will go to the top. You can also put them in chat. But it's best probably if you ask the questions yourself in person, pop your camera on, try to make it as sort of informal and personal as we can. So let's see. I guess I can ask one question. There's a lot I could ask. Could ask about reproductive isolation, sort of thinking about the ecological opportunity and thinking and expanding that to sort of opportunities for reproductive isolation. But I don't think we know that much about the reproductive habitat of these other animals. So of the blackfin form or the shortjaw form. But let me ask about the lineages. So especially from sort of a management perspective. So if we were to, for example, look to, if managers were interested in bringing blackfin back to the Great Lakes, but Julie's work is showing now that it's most likely a different lineage than sort of the Great Lakes being this sort of mixing pot, but now those from Algonquin being of a different lineage. How important could lineage be to thinking about reintroduction, Julie?
In my personal opinion, it's not that important. And in fact, you know, like this is a little puzzle to solve why they're different in Algonquin Park. But in the Great Lakes, it's a mix, but the Atlantic lineage, as far as I can see, is dominating. So that might be why when you use a tree approach, they cluster with the Atlantic. So it might be slightly different in origin, but this lineage apparently has had enough genetic variation and the right genetic variation to evolve into what we call blackfin or nigripenus. So I would think that, you know, functionally they are pretty equivalent to the Great Lakes. The big difference being that they're not used to the real deep lake as the Great Lakes. It's more this part that I would think it would be that we could guess would limit the capacity of these fish to truly live and establish new population in the Great Lakes.
Yeah, great point. And it was interesting too that they actually don't live in the deepest part of the lakes. There's still yet deeper habitat that they could occupy and don't in you know, Cedar and Hogan. Mark, did you want to add anything?
No, all my genetic answers I filter through Julie.
Smart man. Okay. All right, come on. People have got to be ready to ask questions. Don't be shy. Very nice people you're asking.
It looks like Amanda's hand went up, Bo.
Perfect. Go ahead, Amanda.
So really fantastic talk. I enjoyed seeing all of this work compiled into this one presentation. And I wanted to ask where you all feel these forms, sort of to leverage off of Bo's question of could this be used as a source to reestablish populations that fill that same niche. Has there been any work or are there plans for any kind of work that might involve transplanting to see how heritable these differences are within these different lakes and how well those differences might be maintained if you were to move it into a different lake or a different system?
I'll take that one. Amanda, it's Mark here. We have attempted to capture fish from Radiant Lake because of its, we had some ease of access at a particular time. We got several or a couple of dozen to a hatchery, and they proceeded to die over a series of weeks. These are remote systems that are just a heck of a job to get into at the time when they're spawning, which would be in the winter. Cedar Lake has a reasonable road in, but it would have to be plowed for 20 kilometers to reach the lake. So we have tried initially and did get them to the hatchery system where the bloater are being grown for Lake Ontario. Actually, it was in the room next to them, but the bloater didn't help them. So yes, the answer is yes. It's a heck of a job. Since I've been doing this work and describing it in my agency, I've had several people come up to me and say, yeah, that's just like the fish I saw in Lake X over here. And then another one further west of Thunder Bay, Ontario, over there, you know. And so I actually think that there's more of these stories around, and they may present the same story. They also may present better access. Anyways, so we've tried and failed so far.
Okay, great. Nick Mandrak has his hand up. Nick, you want to go ahead?
Yeah. Hey, Mark and Julie. Fascinating talk. I've been following this story all along, and really nice summary of it. Actually, my question is is related to what Mark said at the end about all these other lakes that may have them. So actually in Canada, we're struggling with determining whether or not these are designatable units that should be protected under the Species at Risk Act. And there's two questions that come up that often come up when we're having these discussions. One is, are those regular ciscos just like regular ciscos anywhere else? You know, those typical pelagic ciscos, are they just like ciscos anywhere else? And if that's the case, should we be protecting the ones that occur with the other form? And then the second question is, would you expect in every lake to find multiple forms when you only have cisco, right? And so those are sort of the two sort of questions that often come up when we're trying to determine what these are designatable units, or quite frankly, argue that these are designatable units.
Yes, no, I agree, Nick. So that plot of the Raker density where you had separation of the box plots where the pairs were occurring shows that what we hold in our hand and think as a pelagic, normal, regular old cisco is itself subject to selection relative to the pair that's with it. And that's why those box plots are pulling apart from each other.
Yeah, and that's exactly what we've been arguing, but you know, those box plots would really strengthen that argument.
Yeah. Yeah, they're a unit. So one without the other, and you'd end up with one of those box plots that were red or blue kind of right in the middle.
Yeah. And then the second question, just about, you know, whenever you have a cisco lake, would you expect this to happen? And probably more directly, do you have evidence that it doesn't happen every time?
Yes. In the Northwest, and I mean in the far Northwest, in the lake systems in the Hudson Bay lowlands or near the Hudson Bay lowlands, our broad scale monitoring program comes across cisco populations in relatively shallow lakes, but they're pretty darn cold, right? They're in the Hudson Bay lowlands and they're part of that large wetland system. So in those lakes, you just kind of get your standard sort of cisco that looks a bit like a football. And in other areas, you don't get that. So I don't think they occur. That is not a phenomenon, I think, that occurs everywhere. And I'm going to fall back on our mysis hypothesis and suggest that that's going to be a function of lake depth and mysis diversity. So one of the things that spinning off your question, one of the messages that I would like to convey as a hypothesis is that cisco diversity, I believe, maps onto mysis diversity.
Okay, thanks, Mark.
Great. Thanks, Nick, for the question. Chuck Bronte has his hand up. Chuck, you want to go next?
Yeah, just two questions. The question mark over Lake Superior when it comes to Blackfin. Is that because there were some questionable collections from Keltz's day or some recent observations of suspected Blackfin?
So just a quick reminder for me, Chuck, what did Cal's report about Blackfin in Superior?
Well, I can't recall the details. Probably Randy knows those, but and he's willing to speak up. But I'm just wondering what the question mark was. Is there a doubt that they existed there or not? Is that why the question mark was there?
No, my question mark is actually a setup as well for Julie's hypothesis of a Missourian versus Mississippian refuge.
Gotcha.
No, I just took that from the monograph that, it was questionable if the Negropenis. I'm not going to argue about the taxonomy of the Great Lakes. I just take it what's in the table, and it says that it's questionable, so I put a question mark.
Okay. All right, thank you. Second question is, now I understand, and maybe others on the phone can, or on the call can help out with this. I understand that people are looking at Dave Etnier's old collections from the Minnesota lakes. Is there a chance we would find Blackfin there?
I wouldn't want to say no.
Mark, you led us on this chase when you recommended I talk to Peter Jacobson.
Yeah.
Peter Jacobson talked to us about the Etnier collection, and yes, Etnier does describe Blackfin and many other forms.
Oh, yes, he does. Oh, you're talking about Saginaw.
Yeah, and some others. Yeah. Yep.
Right. So that whole system has the three forms through it, and Etnier used the old Nipigon label for really, the 60 count gillraker fish that are in there.
Okay, and I see Amanda says they have samples from these fish. All right.
Right. So that system, and it's not just Saginaw, that whole system is really, really interesting because they're about 60, 70, 80 plus meters deep, if I'm correct.
Okay.
And that's deeper than the Algonquin Lakes. I mean, a deep part of Algonquin Park Lakes is like 50 meters. So you're at another deeper level, and three-form lakes are relatively uncommon. So I would suggest that system is intriguing as hell from this perspective.
Absolutely. Thank you. And excellent presentation, Mark and Julie.
Thanks.
Another home run.
Thanks, Chuck. Let's go to Trevor Krabenhoff with his hand up.
Yeah, thanks. I really enjoyed that talk as well. Thank you both for presenting. I had a question about trying to differentiate between this idea of parallel origins in situ in lakes, and how one might differentiate that between maybe contemporary introgressive hybridization or patterns of incomplete lineage sorting and ancestral polymorphism, given the relatively short geologic timelines, if you've tried to disentangle between those two. I don't know if that question makes sense.
It's for me. Well, you can talk about introgression if you know that you have two lineages. If you can't have if you don't have a hint to the existence of lineages, you can make story about, introgression, but you just, I'm an empiricist, right? I'm a true empiricist. If you don't have indication of lineages, it's difficult to talk about introgression. I don't know how you do that. And then, like you would have signal of older, more highly diverged alleles somewhere in the data set. And there are some some technique to see if including hybridization in a tree analysis is more likely than not finding. So that's one thing. And second, if you have structure by lake, as I say, and you always have the sympatric forms more related to one another, if you have like two lakes like that, you know, at some point you can still be hesitant, I guess, to conclude. But I think that there is a value in finding this pattern over and over and over and over and over again. So it could be introgression missed everywhere, but when you do find some introgression somewhere and you don't find it somewhere else, I mean, it's at some point I think it's the global pattern that is also speaks some truth. And also, finally, if you find a structure by lake that is sort of equivalent, whether you have or don't have these two forms. So the structure is the same for lake with only one type of fish. I think that if there was something special about, you know, mixing in the lakes where you have two forms, you might have a signal that these lakes are more or less differentiated from the others. So these are the aspect that I would think speak for in situ evolution.
Yeah, thank you. I was curious if parts of the genome might show different genealogical histories, maybe resistant to hybridization potentially.
Well, but that would be interesting to see. But then, you know, that's certainly something I have not done that, so that might be worth checking. But you'd have to check it in many places.
Yeah, thank you.
And the story might not be the same everywhere, you know.
Thank you.
Trevor. Jason Stockwell.
Yeah, thanks, Julie and Mark. This was really interesting talk. Just a quick question, maybe I missed it. Mark, what do you mean by mysis diversity? Could you just talk about that a little bit?
Yeah, no, that's a good point, Jason. And my thinking on this was really the series of publications from Lake Superior and a couple publications from Lake Champlain. And in those are mysis that migrate. Some life history stages migrate. Some life history stages remain near the bottom, and some partially migrate. And it might be even more complex than that. So when I say mysis diversity, I mean their behavioral distribution and possibly life history diversity. So as these lakes get bigger, as I'm sure you know, Lake Superior is quite a great story, and is Lake Champlain, about just how different parts of mysis populations are behaving. And I think those differences are accentuated with depth or a function of depth. And I think, and they have to be stable enough for Cisco, as an adaptive peak, for Cisco to sort of hone in on them, right? There should be mysis on the bottom if you're going to evolve bottom form Ciscos that are exploiting mysis. So I guess I think of diversity very broadly rather than simply purely genetically.
Thank you.
Okay, I don't see any more. Oh, great. Corey has his hand up, and Randy, I'm surprised you haven't asked a question yet. I don't know if you, but let's go to Corey first and then see if Randy has a question.
Okay, mine's real quick, and that was just a fantastic talk. Just speaking of behavior, I was wondering, in the case where ciscos are filling that functional role of whitefish, are there any other noticeable behavior things besides, I think you mentioned being caught towards the bottom, but are these fish, have they been seen like dip into the side to skim off benthic invertebrates or anything cool like that?
No, we have not had any direct observation. White Partridge Lake's just a few hundred hectares, and the south end of the lake is a deeper basin, and these fish are found only in this single small basin.
That's very cool.
The large part of the lake is not deep enough for them. And of course, that's where mysis that's where they hide, right in the darkness down there. So we have not seen them, but I should add that we've had hydroacoustic surveys on all of these lakes, and we haven't dove into that data. And if you do hydroacoustics, it could be intimidating. Anyways, we have 200 kilohertz transducers, and we can tell who's with what in the mysis layers, and these fish remain on the bottom. They do not vertically migrate.
Got it.
There's a whole suite of their pelagic brothers that do.
Okay. Yeah, very cool. Awesome talk, Mark and Julie. Thanks.
Thanks.
Mark, those ones that were on the bottom, those were the Xenithicus form?
Yes.
Yeah.
Yeah.
I did not know, what was new to me that I missed is that there are no lake whitefish in that lake. So that's really
Yeah, it's a
interesting too.
pretty rare finding. You have to go right to the limits of the province to see this. It's an odd situation. Because whitefish is so common across the landscape for us.
Yeah, and as we're just digging into learning more, I don't know if Peter is on the call, but learning more about these Minnesota lakes, it's kind of flipped. Like they don't have a lot of whitefish in a lot of these lakes. They tend to be less common than Cisco. And so,
Yeah.
so I don't know, I think
Go ahead.
all of this diversity beyond the Great Lakes and what we're learning, what you all are learning in Algonquin Park, broader in Ontario, the work that Julie has done before, and then now what Peter and others are describing, Etner is describing in Minnesota. I just think it's fascinating to think about all of this together.
Well, I think that's a good point, Bo, because I mean, what I've learned out of this is that a deep understanding of the Great Lakes is possible by looking around at others, and then realize the Great Lakes is a spectacular case study in diversity, but might be fundamentally different.
Thanks. Wendy has her hand up. Wendy stopped. We're getting an echo. Please mute it if you don't have your phone on mute.
Hi, thanks. Good afternoon. Good talk, guys. I guess sort of just off of Mark's kind of last comments are actually related to the question that I had. So in the Great Lakes, we're starting to see sort of using genetic analyses of early life history of these fish when a lot of the differences in the gill rake are actually come into play. And we're seeing, now being able to genotype, do genetic species identification on some of these, juvenile and larval forms. We're starting to see, fairly early on differences in habitat use that, might be related to the differences in gill raker counts, for example. I was kind of wondering, have you seen any differences? Have you been able to map the sort of the habitat that yet, or do you have any plans to do that the different forms in the lakes where there are different forms are using to see if the different use starts that early?
That would be wonderful. We have no information in larvae. We have caught young of year post-larval blackfin and radiant lake, and they, like this big, right? And they have black fins just a few months into their life. So I mean, the students were fascinated by all the large blackfin that we were catching, and I'm looking at these handful of these, young of year blackfin, way more than these monsters. So it does appear that their early life history is true to form. I think is kind of what you're saying. True to form. Yeah.
Thanks, Wendy. Before we wrap up, Randy, did you have any comments on the talk or questions? You were referenced several times, and your monograph also been really influential. Just wanted to. You're on mute still.
You're on mute, Randy. Sorry about that. That's my fault. Randy, you're still muted.
Nope.
Still muted.
Try it again. How
There
about
you
now?
go. You're good.
Okay. I had some questions, and one was, do you think that last gill arch is functional? That is, they can sweep food from there into the esophagus.
Well, I'm going to have to say I believe it is.
Okay, well, I'm going to take a look next time. I never thought of using it because the theory now on rakers is that on the first arch they don't really strain food. They strain out water, and the food gets diverted along them to the esophagus. So another question I had is that what's your pathway for getting ciscos from the Champlain Sea up to Algonquin Park? Is it the Ottawa River? That's what you're talking about?
No, we're saying, I believe that via, so the argument is essentially there are two movements of Cisco into the proglacial lake basin. From the Atlantic refugium first, from whatever Western Mississippian, Missourian, whatever hypothesis you choose, second. And the Missourian is a better candidate for a two-phase introduction than the Mississippian, given what we found. So we're not arguing for an upstream flow into the park. We're arguing for a downstream flow into the park landscape from Lake Algonquin.
Well, okay. One of the most difficult things to explain in zoogeography for the Great Lakes is how ciscos got into those small lakes in northern Indiana, eastern Illinois, and very southern state of Michigan, because the potential outlets were cut off fairly early.
So maybe, as Julie found for some populations at the west side of Lake Michigan, they may or may not retain the eastern lineage genotype. That's as far as I'm going to go on a genetic answer, because then I run into problems filtering it through.
You know, Randy, that's the part that is not that well explained if you bring the Atlantic lineage in there. That's certain. I think that we need more information about these fish, you know, the genetics of these fish and the genetics of other fish that are found in this area. Because I strongly believe that, you know, people doing comparative phylogeography know that, you know, every species has some, how do you say that, idiosyncrasies, and it's drawing the main pattern across species that, you know, provides stronger indication on where the fish could go when. So I would think that reviewing what is known or getting the data about where do the fish, not only Cisco but other fish in these areas are affiliated to, will provide further answer. But at the moment, I would not say that I'm able to explain how they got there.
Thank you.
All right, so a comment from Nick. Don't forget about the diversification of lake whitefish and lakes that don't have ciscos. Very cool. That's a great point, Nick. Also a comment about, I don't know what this is referring to, ice edge proglacial lake.
Bo, that's just a potential explanation for those areas. And Chris Wilson and I covered that a little bit in our lake trout chapter in the recent lake trout book.
Okay, to explain those Indiana.
Well, just in general, how, you know, we can't forget, like, when you look at the dike map, you can see the post-glacial lakes there, but keep in mind that there was lakes, water pooling at the edge of the ice as well that isn't often well mapped out. Well, is usually not mapped out in these post-glacial post-glacial maps that are produced. And that those were very dynamic systems compared to like these major water bodies.
Yeah, agreed.
Well, I'm really grateful that so many of you all are thinking about the glaciation and providing some things for people like me to learn. Never thought I would be thinking about all of these things when I started studying fish ecology a long time ago, but certainly relevant to understanding lineages here, colonization. So I think we have hit a good stopping point. Again, just for those of you that have remained with us, and it's still 53, remarkable. Again, just a testament to the quality of the speakers we had today and the quality of their presentation, and also the interest in these fascinating animals. And so thanks again to Julie and to Mark for a wonderful talk, and I hope you all will join us again next month for what we hope will be another great talk. And we'll get advertisements out on that in about 2 weeks. Thank you, Carrie, for hosting us, and everybody have a great weekend.
Thank you, everybody.
Bye.
Thank you.